Okay, I think we're ready to get back to our regular programming. I want to introduce Erin Middlebrook. Erin is the BDB Resident Applications Expert for the BD CellView image technology and a longtime flow cytometry user. He has spearheaded all data generation efforts to evaluate and market BD CellView image technology. Erin is also well-versed in high-parameter flow cytometry with over 25 years of experience designing and running panels in both traditional and spectral flow cytometry. Prior to his work on application development for the BD CellView, Erin spent several years working in reagent R&D, where he successfully established a tumor dissociation workflow for flow cytometry. Erin's graduate work at the University of Arizona and his postdoctoral fellowship at the Gladstone Institute of Virology and Immunology at the University of California, San Francisco, was focused on T-cell development and function within the context of autoimmunity and HIV. Erin, welcome to FlowTex.
Well, thanks so much for the introduction. I need to rewrite that. It's fairly long-winded. At least it feels like it when you're sitting there. Oh, nice. So yeah, first time at FlowTex. It's such a cool meeting. I got to come here a year ago, maybe a year and a half ago, to place the S8 in Joel's lab, and really enjoyed Houston. I'm not getting any breakfast tacos this time, so I'm a little upset about that. But maybe there's still one more day that I can check that out. So today I thought I'd talk about combining spectral and imaging together, just because I feel like that's one of the real strong suits of our platform, that we have the ability to look at both kinds of experiments within the context of imaging or spectral. This is actually data that I covered in a webinar, but it's such a tremendous amount of data that I really had to do a gloss over. I kind of turned it around a little bit and tried to talk a little bit more about panel development and how I use, what tools I use to find whether or not I'm going to get good imaging in the context of my spectral. So I'm going to go ahead and get started. What do I do? Click. Arrow. Okay. Yeah, so now we have to have this disclaimer up. We are Waters now, although I'm still trying to figure out how that works. I looked at my title slide, it said BD Facts Discover. Do I go Waters Facts Discover? So right now I'm just Facts Discover applications. So we'll get there. Yeah. So far nothing in my life has changed, but I'm sure there will be little and big things on the horizon. I always like to start with just the most basic part of the imaging, which is the mouse over feature, which for those of you who've sat on a S8 or A8, you know what I'm talking about. For those of you who haven't, it's really cool and magical, and it happens like instantaneously as you're looking at your plot. You can just mouse over any image you want and see exactly what's there, whether or not it was debris or a schmutz, or if it's a cell, a big cell, little cell, doublet, clump, whatever. It's going really fast. Anyway, I usually don't go this fast. I think it's playing at like 1.5 speed, maybe two times speed. But the idea being, you have the ability to control what the image is, what the image is. If you want to look at bright field image or your fluorescence or scatter image, you can control that. So in actuality, it gives you, the ability to look at your sample in 5 dimensions, because you've got the dot plot in 2, and then up to 3 or even more colors on the plot. But let's talk a little bit about what those images are. Oh, I can't use that. Okay, so here's the image that we kind of ended on there. And this particular image, what it looks like is it's a light loss image, which is the gray in the background, and most of the other structure of the cells obscured by the fluorescence. And then we have a nuclear stain in the red, and then we have a green stain, which is a surface marker. And this happens to be a cancer cell stained with FITC, EBCAM, and DROC5 in the nucleus. Now, each event is not just what I have here. Each event actually consists of, in this case, 5 different pictures. And I'm choosing to only look at the 3 at one time, but I could look at all 5 at the same time if I wanted to, but it gets nonsensical after you have about 3, I think. So in this case, I have 3, and it's a 2-color fluorescence experiment, so there's only 5 available, but there is another fluorescence available too. So we have the light loss, the side scatter, the forward scatter, the DNA, and then the surface marker. Now, the images are fantastic, and they give you a lot of insight into your sample. As you get more kind of familiar with using these, I actually found that it's a lot like a longtime cytometrist and how they can dissect a scatter plot and tell you exactly where your unlysed red blood cells are and where your clumps are and where your debris is, and they can tell how good your sample prep was as soon as they see that scatter plot. And it's the same with the images. As you get more and more used to looking at them, you can see the darkness of the cell membrane oftentimes corresponds to the health of the cell. If you have really old cells or fixed cells for a long time, that cell membrane starts to deteriorate, and you can see it instantly. And you can see if somebody didn't wash it enough, or if they had a really tough sample prep, or if it's like a tissue and it's like, obviously a shotgun scatter, you can see all that debris and why it looks that way on the scatter. But that's all what I call like the qualitative level of the imaging, where you can just use it intuitively. And then the second layer to that onion is obviously we have all these imaging features. And these imaging features are measured in each of the 6 channels, and they can be plotted just like MFI or fluorescence can be plotted. So in this case, we have a plot on the far left of max intensity versus total intensity of the light loss. In the second one from the left, we have a side scatter plot, and then a forward scatter plot, a DNA plot, and a surface marker plot. So you can actually pull apart the biology of each particular event using these image features. So in this talk, I'm not going to talk about all the imaging features. It ties up a lot of time, and I've done it so many times, and it's online, and it's everywhere. But for this one, I'm going to talk about the imaging features that I use in this particular application. But needless to say, they can be quite powerful, and I think there's a total of about 13 imaging features per channel that get extracted. So if you have a 3-color fluorescence experiment, it's something like 96 imaging features or 107, depending on which fluorescence you're using. So for the assay today, I wanted to talk about the morphology of activation. So one of the things I noticed, I was doing a activation assay probably about a year ago, and one of the markers I had in my panel was CD54. And what I found was that upon activation, CD54 seem to have a lot of different flavors of expression. So if you look at those collection of cells up there, you can see in the top left, you have a cell that's I can point this. In the top left, you have a cell that's kind of uniform, really bright expression, and there's several of those here. You can also see some that are very dim but uniform expression, and then you have these other flavors where there's a puncti of expression on one side of the cell, and in some cases it's very small, and and then there's some that it kind of engulfs one whole half of the cell. And so it was very curious to me, and I was looking at several different activation markers, and it wasn't all of them. A lot of them would just go from being uniformly dim to uniformly bright. And then I had some that were just always started as a puncta and then polarized and then uniformly bright. And it was curious to me because I hadn't seen it before. And then once I saw it, I started looking for it more, and I started seeing it more. And so what I found was that there's a particular collection of image features that you can look at. So in this case, what we're looking at is a plot that's showing the distribution of CD54. So we have size on the y-axis, and on the x-axis, we have delta center of mass. So center of mass is the measurement, and I'll get into it in a second, actually. So this is the center of mass, the delta center of mass between side scatter and CD54. And with this plot, I can actually pull all 4 of those phenotypes out. So in the top left, I have uniform expression, uniform bright expression. The bottom left, dim uniform expression. And then as you move to the right on this plot, you see the signal becomes polarized in the cell. So in the bottom right, it's polarized and very small. Top right, polarized like half the cell. So how does the imaging feature do this? What it's measuring is, if you look at this collection of images, on the top, you have one event on event A on the top row, and then event B on the bottom row. And then in the far left column, you're looking at light loss in CD54, side scatter in the blue, and then CD54 alone. And so what it's measuring is the center of mass of one channel. So in this case, the center of mass of side scatter, which happens to be approximately the center of the cell. And you could use light loss for this, or forward scatter likely, anything that captures the whole footprint of the cell, and you look at the center of that, it's roughly going to approximate the center of the cell. And then when you look at the CD54, when it's uniform, it's pretty much the center of the cell as well. So the delta between the center of mass of side scatter and the center of mass of CD54 is approximately 0 on a uniformly stained cell. Whereas once that CD54 signal shifts to one side of the cell, then the center of mass obviously shifts as well. And so you're actually measuring that difference. So in the top, the delta between the two centers is effectively 0. And on the bottom, the delta between the two centers is a bigger number. So it's not a huge distance, but on a plot, you can actually capture that. So as this delta center of mass goes up, this thing's changing on its own. Anyway, as that center of mass goes up, the signal is polarized, and you can actually capture that using this imaging feature. So for the experiment, what I wanted to do was I wanted to do two things. I wanted to really showcase the power of our spectral technology and how much you can pack into one sample. And I wanted to couple that with some cool imaging. And so I decided to try and go after these polarized morphological kind of aberrations, or whatever they were, to see how frequent they are and when they occur. So to do that, I used the stimulation model. So I have 6 different conditions here. So I stimulated for 24 hours and 48 hours. I stimulated with poly-IC, which is a kind of an innate stimulus. It's a TLR3 agonist, so it stimulates the myeloid arm of the immune system. And then I used anti-CD3 and anti-CD28. So I didn't use bead bound. I found that the bead bound dinyl beads are so robust. Everything just either starts dividing and goes crazy so fast, and then you start to get a lot of cell death. I actually prefer the plate bound version. So if you incubate a plate for 45 minutes with CD3, it actually adhere to the bottom of the plate. You can put your cells on top, add some soluble CD28, and you actually get a little bit more of a gentle stimulation. And so I used the one on the right for this. And the idea was, okay, we've got these 6 conditions. Oh, I did this with PBMCs. The idea was, okay, we have these 6 different conditions. How can we effectively multiplex those into a single sample and look at a variety of activation markers within the context of a pretty basic immunophenotyping panel? So in order to do that, I generated a panel. And what you'll see there is I've got a bunch of T cell markers, a couple B cell markers, some NK cell markers, and then I'll get into what the imaging was at the bottom. But then the other thing I did was I used CD45 in 3 different flavors to try and barcode all those 6 conditions into a single tube. So I would stain with the backbone all together. So it's a 21 color backbone. And then what I did was I had a wild card, so a drop-in and RB705. So the drop-in was all these activation markers. So I have 2 imaging channels, or 2 imaging targets here. I'm using MitoTracker. So MitoTracker stains the mitochondrial DNA, and you can actually use that as the surrogate for activation as the cell goes from being a quiescent, naive cell to an activated cell that's mitochondrial footprint actually enlarges quite a bit, so it'll expand, become a lot brighter. And you can pick this up on traditional cytometry as well. But I find that it's a really good surrogate on the imaging, so I can look at, it'll tell you the size of the cell and how activated the cell is. And then on the bottom there, I have the RB705 drop-in. So I just basically went through our entire catalog and pulled out any activation marker that we had in RB705, which is a fantastic imaging floor for channel 3. And the idea was we're going to put that in each well of a plate. So again, you got your 21 color background, and you have the wild card or activation marker in each one of the wells in that plate. So before I shift into what the results look like and how I found all the ones that were polarized and not polarized, I wanted to talk a little bit about panel design for this. So this is a pretty large spectral panel to also include imaging, but for this system, you have to kind of think about it in two ways. So there's obviously there's the spectral part of it. So you have all these APDs and a very elaborate and sophisticated setup in QC that enables really fantastic spectral resolution. But then at the same time, you have more of a traditional approach with the imaging, where the imaging is done on PMTs. And there's three channels. So there's an imaging channel, channel 1, which is basically equivalent to FTSE, channel 2, which is equivalent to PE, and then channel 3, which is more like the PE size 7 range. But these are all done on bandpass filters and PMTs until still there's no compensation. We have compensation coming in 1 month. I'm so excited. I've actually been using it for about 3 months now, and it's a game changer. It's fantastic. So I can't tell you enough about that. I would talk about it more, but there's not really time. Anyway, so you've got the imaging off 6 channels, and then they're all tied together with this event ID. So the event ID is here, and all the imaging features are measured with these onboard algorithms that exist in the firmware and through FPGAs, and we can tie all that data to that event ID, which makes it sort compatible. Here's some example at the bottom of all the images we have there, and then the numbers for the actual imaging parameters. Where there's 96 total that get pulled off in a 3-color fluorescence. So when you're designing a panel, these two things don't exist in a vacuum. They coexist. So you have to think about how much my spectral dyes are going to impact my imaging dyes, or my imaging, just my imaging period. So that's kind of the biggest challenge you have in designing a panel for both imaging and spectral. And so all the other rules of panel design apply. So you design a panel as you normally would. High density antigens use dim fluors. Low density antigens use bright fluors. You need to consider co-expression and or exclusion, whether it's a dump gate or if it's widely expressed across your whole panel. And then here's the critical one for this, which is you want to choose fluors with minimal secondary excitation of the blue laser. So you have to know where your imaging fluors or your imaging targets are going to fall and design your panel accordingly. So the way that I do that is I love these spectral plots. So here's my panel down here. I've got a total of 22 markers. And I just kind of systematically go through. And what I'm looking at is that blue area, right? That I can use this. This kind of blue area. I'm trying to look at, obviously I have blue specific dyes that I'm using for imaging, which are the Mito Tracker and the RB705. But what I'm really worried about is the secondary excitation from UV dyes, from violet dyes, from red dyes that kind of will accumulate in those regions and give me background fluorescence that's going to confound my imaging. And so the way I do that is I kind of go through this, and you can start to see, if you look at these, these bumps down here, the values on these are normalized. So it's taking the brightest peak and making it 1. So it doesn't represent how big that bump is in the blue. So it's a little misleading because of how small these are, but I'll show you how to get around that. So we go through our dyes and we build this panel, and we can see, we're packing the UV, we're packing the violet. I think this is maybe the biggest imaging panel I've ever done, the biggest spectral panel with imaging I've ever done, but it's only 2 imaging channels, so it's kind of a cheat. So as we build this, we can see the blue is obviously pretty empty, but you can see there's some accumulation of signal that's happening in the blue. I'll put the filters in a second, and you can see which channels it's impacting the most. But as we pack this, you can see everywhere else is pretty full. And I stay away from yellow-green dyes because yellow-green dyes happen to usually have pretty big excitation on the blue. So I've minimized my yellow-green dyes here. So I've packed the red, I've packed the violet, and I've packed the UV. And so what you end up with is, your 22 colors, And now once I put this up here and I can see where my filters are, I look like I've got some signal, major signal in imaging channel 3, a little bit in imaging channel 2, and imaging channel 1 looks pretty clean. So again, I'm using imaging 1 for MitoTracker and imaging 3 for RB705. So mostly concerned about that imaging channel 3. So what I do is I run an imaging FMO, which is essentially all the dyes that are up there right now, minus the imaging ones I don't put in. And the next step is I run an unstained control. So this is looking at unstained cells on the top, and then my imaging FMO on the bottom. And so I run unstained cells and I draw a gate where, and this is imaging detector 1, total intensity, imaging detector 2, total intensity, and imaging detector 3, total intensity. And the gates you're seeing were drawn on the unstained cells. And so what I'm looking for is, with that imaging FMO, any sort of migration outside of that gate. And so there's multiple ways you can look at it. You can look at it on a histogram using this approach, and then you also want to look at your imaging, right? So you want to see, is there any stray fluorescence in my imaging? And what I found is that the histograms are by far the most sensitive, whereas the imaging, because of your ability to adjust the minimum and max, you can hide a lot in there. And so, what I do to mitigate that is, if you look at this histogram, this is like your max and minimum intensity controls on the instrument. And this, you can see there's a positive signal here. This is my CD3 BV515 control. And this is kind of where I would have my min and max regularly. So what I do on these, the unstained and the FMO, is I actually take that min and I drop it all the way to the left, and I tape the max and I put it right about in the middle of the background. And what I'm trying to do is find the noise floor. I want to see if the noise in the sample is totally random or if it's cellular in nature. So if it's random and it looks like this, that means the signal that's in there is not because of a cell, it's just inherent in the system. Whereas you can see in this bottom one of the previous iterations of this panel, it might be hard for you to see there, but there is a shadow of a cell there, right? So you can take those and you drop them, and so that's the spillover coming from those non-imaging floors. And if it shows up as a cell, you know it's real, it's coming from a cell, and it's going to confound your imaging features. Whereas if it's random and salt and pepper-like, you know it's just in the system and it's not there, right? So it's not going to impact the imaging features. It's not going to show up in your ROA when you set your ROA, and it's not going to confound your imaging features. So that's kind of the key to doing this, is finding these little tricks to really interrogate the system to find out where the signal's coming from and if it's going to impact, whether or not your imaging features are measuring true biology or spillover in this case. And it's one of the big problems with this technology, is that we don't have the ability at this point to compensate spectral dye spillover into imaging. So it's really a panel design issue. And I've talked ad nauseum to the math guys, and they're like, yeah, every way they do it, because the imaging information is there, but you have no way to do it on a pixel basis. You would have to do a uniform correction, and then it wouldn't be specific to the spatial distribution of your cell. So it's confounding. So it's a critical part of combining these technologies, is to be able to troubleshoot your panel. Oh, and this is cool. We just did this fluorochrome guide. I was supposed to plug this, and I'm going to plug it. This is awesome. I worked really hard on this. It's a huge poster. We just came out with it, and it's probably going to be available, I think, electronically this week or maybe next week. And then in conferences and at Cyto, they'll have printouts for you guys to grab. But it's really big. It's like 3 foot by 2 foot. It has a whole imaging thing down here at the bottom that shows all the imaging features, and it has examples of all of our imaging floors. But more importantly, it's got this fluorochrome guide. In this case, you can see what it's showing you here is it has what the emission of a particular dye is, what its relative brightness is, and then its spectral spillover is here. So if you were to look at these, this corresponds to this, and once you start to use it and you look at it a little more, it's really useful. And then it also tells you what the impact to each imaging channel is. So BV711 is not so good for imaging channel 3, whereas BV650 has no impact in your imaging. So it's a really nice tool if you're going to use this instrument for panel design. So let's get back to the experiment. So once I had my panel was troubleshot or trouble-free, I ran this through a couple times and I started to go through the results. So the barcoding worked really well. We have a barcoding kit that we use, but it's intracellular and I don't like that. So I kind of came up with a surface method and it worked really well. So this is using CD45, and with just 3 floors, I could multiplex all 6 conditions. So if you look at the little layout here, you're looking at double positives and single positives, and you're able to get all 6 of these samples multiplexed into the one. So then from there, I went through and I developed a little gating strategy here. And the gating strategy is mostly a few populations of T cells, a few populations of B cells, and then some NK cells. The total of 15 subsets I was looking at. But if you start putting all this data together, you've got 6 different conditions in each well, and then you've got 30 different wells for each of the activation markers. It's a tremendous amount of data. So I had to figure out a way of analyzing this data that was a little easier than going through dot plots and doing mouse overs. So I came up with an approach for that. But as you go through these, what I found was that I hadn't had any experience with poly-IC before. Poly-IC is apparently a really fast-acting thing, and so I missed most of the poly-IC activation. I did catch a little bit, so it was mostly focused on the CD3, CD28. And as you go through these, and there was some panel optimization for this too. I really didn't want to include markers that are traditionally used for phenotyping like HLA-DR because it's so impacted by activation. So I was going to have massive changes in activation. I wanted to have a gating strategy that was basically untouched by activation so that I could compare specific populations, right? So my gating strategy only used markers that remained unchanged before and after activation. So it may seem a little weird in terms of some of the populations I went after, but that's why I intentionally left those off, because I wanted to see how HLA-DR reacted, or how 69, or some of these other markers that are sometimes included in these immunophenotyping approaches. So if we focus on one population here, I've got some CD57 versus NKG2D NK cells. And so when I go in and focus on those NKG2D positive, CD57 negatives, what I found was, and that was the other part of this too, was because they're multiplex. So I was acquiring, I think, around 250,000 cells per well. But once you unmultiplex and then you go into specific subsets, all of a sudden your numbers are very small, unfortunately. So the radial moment looks a little sawtooth, a little jaggedy. I really like those more smooth histograms. They give me more confidence, but we're going to go with it. So this is what the mitotracker actually looks like. And you can see the ones on the top have that really small green mitotracker footprint, whereas the ones on the bottom have this kind of more robust, and that corresponds to the energetic demands of activation. So as these cells begin to start to proliferate, their energetic demands go up, their mitochondrial footprint goes up quite a bit, and you can actually just clearly see it. And that's the other part about the plate-bound activation I really like, is if you use dinyl beads, everything just goes nuts. Everything looks like the ones on the bottom. Whereas if you use the plate bound, you end up with these early activators. Half your cells are activated and half your cells are not activated. So you see a clear difference, and you see a clear behavior difference in the cells as well. So then what I looked at is looking at that same plot. So I've got that, I've got delta center of mass between side scatter, in this case CD18, and on the y-axis I've got size. And what I found was that there were very clear differences in the way that these plots looked, and those corresponded to basically 4 different phenotypes, where you have non-detectable CD18 expression, very punctate condensed expression, and those existed in the unactivated, the unactivated, and then once they started to activate, you got the more polarized and then the uniform expression. So I really leveraged this plot between delta center of mass and size and applied it in more of a uniform way across the whole sample. And so this was kind of my pipeline that I used. Was I went through and first I concatenated everything, which is kind of a bear. So that was 30 wells at 250,000 events each. the concatenated UMAPs and TISNEs were awesome though, because then I could go through and I could gate on my mitochondrial high radio moment cells and see where my activated cells were in each population for each stimulation. I could see where my expression of my drop-in marker was happening. Was it on my B cells? Was it on my T cells? And I could look at those things in parallel. So eventually what I ended up doing though is I started to generate these batch reports. So the batch report is actually predicated on the shape of this that one plot. So this is again looking at that delta center of mass between side scatter and RB705. And what you see here are 4 very different profiles, right? So the one on the left, we have B cells, HLA-DR, 24 hours, CD3, CD28. And we see all the cells are kind of in that top left quadrant. For ICOS, CD3, CD28, you see more of a smear, but it's all on the left side of the plot. Whereas for ICOS on B cells, it's completely shifted over to the right side of the plot, but it's a smear. And then for the NK cells on CD54, it's kind of everywhere. And so what this corresponds to are, there we go. So the ones on the left there are really uniform, bright expression. And then for ICOS, it's actually a range, and you can see the cell size changes too. So the cell size on the bottom, and you get really dim but uniform expression. And then as you move up, it gets into that bright big cell. And then for B cells, for ICOS, it was the craziest thing. It was almost totally exclusively polarized. But it changes in size. So the one on the bottom right is very small and punctate, and then as you kind of go up, it engulfs the entire half of the cell. And then with CD54, I had all 4 of these phenotypes present. So what I did was I went through and I developed this kind of basic analysis metric. So I have 4 sets of plots here. The one on the left is looking at the radial moment of mitotracker. So that is an indicator of activation. So I could quickly look at this and go, okay, in my unstimulated sample, I had a mitotracker of blah. With poly-IC, a mitotracker did this, which is the same as untreated. But then with the CD3, CD28, we see a shift to the right. So I know T cells got activated at 24 hours. And then I can look at the CD58 expression in both of those. So the unstimulated and poly-IC look the same. And in the CD3, CD28, I have this shift to the right. So I have increased expression of my activation marker, and I have activation. And then I go ahead and I plot this at 24 hours, looking at poly-IC and CD3, CD28. And once you start looking at these plots, you start picking up the patterns really quickly. You're like, oh, everything's shifted to the right. This one's polarized. Cool. Everything's shifted to the left. It's uniform. Everything's in the top left. It's very bright. And so you get to kind of quickly scan these. And so I did one of these for every cell type, which was kind of insane, and every condition. So this was 1 page of craziness for CD58. I've got T cells, I've got B cells, I've got NK cells, I've got monocytes, and then I've got 24 hours, and then I've got 48 hours. And that was a lot, but I only had 30 to go through. So I was quickly able to scan through 30 and be like, oh, this one's good, this one's good, this one's good. And it really gave you a quick way to see, how much activation I was getting, how much increase in activation expression I was getting of that particular marker, and then what the morphological kind of spatial distribution of that expression were. So then I had to go through all 30 of these, which was not trivial, but it was actually kind of fun. And it's so cool too, because you're assessing something that's measured on an image, but you're using the FlowJo kind of ecosystem and stuff you've been doing forever, batch reports, putting them in binders and getting a highlighter and getting a cup of coffee and going to it. So what I found was that out of the 30 markers, there were 24 that showed increased expression after activation, whether it was poly-IC or if it was CD3, CD28. And of those 24, they kind of distributed to different cell types. So in this case, you can see I've got T cells in green, I've got NK cells and B cells, And then those are the 24 markers that showed an increase in expression after activation. And some of them were common to all 3 cell types. So I've got a collection there that showed increased expression in all 3 of those cell types. I've got some that were specific to NK cells or B cells, and then a handful that were specific to just T cells. And so then out of all those, what I found was there were 9 that showed kind of the hallmarks of that polarized phenotype. And the interesting part was, and I think you saw it in the slide I just showed, where you had, I think it was the ICOS. ICOS in T cells was uniform, but ICOS in B cells was polarized. It was population specific or it was activation specific. So it was only in poly-IC or only in CD3, CD28 that I see the polarization, and it was only in this particular cell type. So whenever I see that things are unique to a particular subset of cells, it feels real, it feels biological, it feels non-random. First, I thought it was just maybe one of the stages of expression, that everything starts out punctate and ends up uniform. And we never saw it before because we were just looking at positive versus negative. So something that was dim was punctate and something that was bright was uniform, and that it happened to all markers. But that wasn't the case. I saw that dim uniform and the punctate in the same marker depending on which cell type I was looking at. So I'm curious to see where this goes. The next step for me is, yeah, I already said that. So the next step for me is now I got to put it in our pipeline. I want to put this on the S8. I want to sort these, and then I'm going to do some, whole transcriptome, Rhapsody stuff as soon as I get my budget approved. And I'm going to try and track a gene down that is regulating this. That would be the ideal. Ideally, I'd like to tie it to a function. That would be kind of the holy grail, is when they have this punctate, they can't do this or they can do this. When they, but first I gotta try the genomic expression part first. So the first step we did transfer it to the S8. And this is looking at CD54, and we sorted them onto slides. And you can see it's probably not the greatest lighting in here, but you can see those ones on the top left are obviously super robust expression, uniform. And then the ones on the bottom left are going to be kind of more dim uniform. And then the ones on the bottom right, you can see it's one little bright spot. And then on the top right, they're kind of more polarized versions. So I'm not measuring artifact. It's a real thing. And next step is to dump these into Rhapsody and see what kind of genes are regulating this stuff. I think that's all I have. Oh, and this is just kind of the summary slide. So there were 5 that went up with the poly-IC and 23 that went up with CD3 and CD28. And we'll see where it goes. So stay tuned. I'm supposed to plug this Horizon event. So I'm coming. That's one of the things I wanted to talk about, my latest work, but they told me I can't because I have to talk about it at Horizon. So I get to talk about that on March 17th. I think it's our second stop. We're doing one in New York City, and then we're coming to Houston on March 17th. So stay tuned. I hope to see some of you, because I told them no one would go to both, and I should be able to do the talk at both venues. And they said, no, they're all coming. So we'll see how that goes. But thank you guys so much. I hope I didn't go over time. Thanks.
Thanks so much, Aaron. Do we have any questions for Aaron right now?
Aaron, thanks. That was great. I had a quick question about, and I might have missed this, but as you were going through, you kind of did a gating strategy based on, hey, I made this observation in the imaging, and then I'm going to dig down and ask questions based on that. So now what if you take your whole data set and hand it off to somebody that doesn't know anything? Do that same clustering using the imaging features. Can you now kind of get to those separations of those populations? So now you're saying, hey, what's going on with my activated cells? Do you then get those phenotypes that are being pulled out using the imaging features and automated clustering algorithms, et cetera? Do you have that?
So I think that you get the activation shows up for sure. I saw that myself in the clustering that I did. Right now, I think that the polarization part didn't, for whatever reason, and I think it's because it's not consistent enough across the different markers. If you looked at each one of them had its own little unique distribution. Whereas I think if there were like 4 or 5 that had one distribution and everything else had another distribution, it would come out. But in the UMAPs and TISNEs, it just kind of looked like a schmutz. I didn't get defined kind of populations, and then maybe just be a numerical thing, right? Where, because I only had 9 out of 30 that showed polarization, and that polarization was unique in each case. In some cases, it was all punctate. In some cases, it was half the cell. So, but more to be done there for sure.
That was a really nice talk. 2 questions for you. One is, it seemed, I wasn't sure. So when you first do the image analysis, you have all these different characteristics that you can look at, right? And then those are what you can also put into FlowGeo later to then do the batch. And then can you export the images completely out and still keep that Innovate ID in case I want to do something like what EM does for class averaging or do more traditional image analysis segmentation?
So the cool thing about the way the system is set up is all the imaging features are, numerically calculated in real time so that they're sort compatible, and they're packaged in the FCS file, right? So if you just looked at the FCS file, you'd be able to do UMAP or TISNE analysis using those imaging features. And then if you found a population you really liked, you could actually go track those image IDs down in your FCS file, and you could go back and get the images. So you really can get a lot of benefit from it and never touch an image or always have them there. So, and you can export them. You can export it just FCS or FCS plus imaging. It comes out as a FCS file in this other file type called the CVW, which is a cell view waveform file. It's just the waveforms. And then that unpacks in stacked tips, which is compatible with, Cell Profiler and whatever else you want to use. So it's a nice kind of marriage of the two.
Thank you so much for your talk. My question is, so you can track I would say, like polarization of like certain organelles like the mitochondria and immune cells, but can you also test that, I don't know, in more polarized cells like epithelia or, more structured cells? And I don't know if, is that also possible?
I mean, the short answer is yes, but the longer answer is that, those cells tend to behave differently in suspension. So once you get them in suspension, they tend to ball up, and, sometimes the biology gets lost. They're not doing what they were doing when they were on the bottom of a flask when they were happy. So if it's robust and it withstands that transition from flask bottom to suspension, then it'll show up on the instrument. It's just a question of, whether or not that will persist.
Thank you.
Thank you so much.
Cool beans. Thanks, everybody.